Hello, friends. Welcome back to another discussion. Today, we will discuss about virus. The topic of discussion will be the basics of virus, the viral replication methods, and the various methods of viral cultivation, and also how to quantitate the viral growth. So, these are the topics we have to cover today. So, let's begin with a basic introduction on virus. Viruses are obligate intracellular parasites. That's by definition virus. And they lack, usually, they do not have uh, a machinery or a required materials for the protein synthesis or nucleic acid synthesis. These two are considered as the most required processes for a living organism. And hence, these viruses do not have these capacities. They sometimes considered as non-living particles also, because when these viruses are outside a host cell, they do not show any features of a living material. They simply act as a particle outside a cell, and when they enter into a living organism, they shows the properties of a living feature. Here, what actually they do is they hijack the machineries, the protein synthesizing and the replicating, that means the nucleic acid synthesizing machinery of the host system. And by utilizing the enzymes, proteins, and other structures of the host cell, the virus will synthesize their own proteins and their own nucleic acids. And thus, new viral particles will be produced. And these newly synthesized viral particles released from a host cell are now called virions. These are the freshly released viral particles but not attached onto a host cell, are called virions. So usually, for the cultivation of viruses, hence these are obligate intracellular parasites. We cannot do this cultivation simply like in case of a bacteria, where the cultivation is easy. We can provide a nutrient medium. But here, in case of a viral cultivation, we need to provide a living cell as the substrate. So the cultivation usually occurs either in tissue culture or in embryonated egg, or also it could be done in a living host. So these are the basic methods for the used for the cultivation. And when you go to the structure and other details, these are very tiny particles, very microscopic, and not observable through even through light microscope. You need a advanced type of electron microscopy, and all. They have a range of 20 to 300 nanometer. It's much smaller than a bacteria. When we see the structure of a virus in detail, there are basically two structures. One is a genetic material, the other one is a protein coat or a covering of protein, called, usually we call it as a capsid. This capsid is in turn made up of smaller units called capsomeres. So in certain viruses, in addition to this capsid, there will be an external covering, external membrane envelope called or external covering called an envelope. And this is usually made up of lipid material, or it's having a lipid in chemical nature. So these are the three most important structures seen in the virus particle. There will be a nucleic acid, and this nucleic acid will be either DNA or RNA, and there will be a capsid. Capsid is nothing but a protein covering over the nucleic acid material, and above which there will be a lipid material, call it as an envelope. So this is the basic outline, basic structure of a virus. And based on the symmetry, based on the shape of the capsid, we have different classes of viruses, like a helical virus with a helical capsid symmetry. There will be polyhedral virus. There will be spherical viruses. There will be complex viruses. Okay, most of the bacteriophages coming under the complex symmetry. So in complex symmetry, actually there will be a polyhedral head and a tail region, and the other materials are also there. That means like the nucleic acids, and also there are tail. There'll be some attached structures in the tail region, and additionally there'll be the external receptors like spikes and other specific receptors to interact with the host cells, also seen in a viral particle. They are usually coming as a part of the capsid. So these are the different classes of viruses based on their capsid symmetry. And based on the nucleic acid, we have two categories: DNA viruses and RNA viruses. And within DNA, we have single stranded DNA and double stranded DNA. You can find different examples in this table. In RNA also, we have single stranded and double stranded viruses. So these are some representative structures for understanding the uh, structural details of viruses in general. You can see the Ebola virus, chikungunya virus, influenza virus, and the basic structure of a DNA virus. So all these are given for your understanding of the structure of virus. And based on host, the viruses are again classified as bacteriophages. When certain bacteria infect a bacteria, that means they require a bacteria for the survival, then such viruses are called bacteriophages or bacterial viruses. We have different examples like T phages, P1, lambda phages. All these are different examples. They infect the bacterial cells. There are animal viruses. Definitely they attack animal tissues. Okay, most of the human pathogens are animal viruses. We have various examples for that also. Then plant viruses. These are most of these plant viruses cause plant diseases. We have various examples like tobacco mosaic virus, CAMV, potato virus. Various viral diseases are there. So they attack on the plant cells. Then cyanophages represents that those which infect the cyanobacteria, and mycophages are those infect or those living on fungi. So these are different classification based on the host requirement. Then you can see the bacteriophage structure. This is actually these are the different representative structures of bacteriophages. You can see icosahedral structure. Also you can see the head and tail, that means the complex symmetry structure. Also you have a filamentous type of phage. So all these phages are possible to infect bacteria, and in which this complex symmetry is the most widely used structure as a bacteriophage structure, in which you can see there is a DNA or RNA molecule inside the head region, and the head is made up of the capsid protein, and there is a neck, and below that regions below is called the tail portion, and the tail portion is covered by a tail tube and a tail sheath, right? And there are tail pins and tail fibers. So this is the basic structure of a bacteriophage. And coliphage is a term usually used to represent those viruses which infect E. coli, and most of the T phages are coming under this category. When we talk about the general phases of viral replication, as already stated, these are obligate intracellular parasites, and they do not have any replication machinery or any enzymes or proteins required for their multiplication. So how they do this process is, as already stated, they they utilize the machineries and enzymes and proteins from the host system. So for that, they need to enter the host cell first. So the replication process begins with attachment. So the host cell need to attach onto a the viral particles need to attach onto a host cell, and this attachment is facilitated by the presence of the receptor proteins, usually as noted, like the spikes and other receptors on the viral particles. Then the second stage will be entry or the penetration. Usually, not the entire virus enter into the host cytoplasm. Instead, the genetic material or DNA or RNA is being injected into the cytoplasm. This is what we call the entry or penetration, or it's the injection of the genetic material into the host cytoplasm. Then the third process will be the replication process. Actually, in between that, the cell either lies the host system, or it enter into the host chromosome, and they exist as a prophage kind of system, especially in bacteria. So anyway, after entry of the nucleic acid, after entry of the viral chromosome into the cytoplasm, they replicate. They replicate the viral genome, and that will be copied. And also there will be expression of certain viral genes with the help of the host transcription translation machineries. So certain genes are expressed which are very essential for replication and the growth of virus, and also the virus will take up the replication system of the host cell for the synthesis of its own genome. Then after that, once all these materials have been synthesized, that will be synthesized, like nucleic acid is synthesized, then the protein is synthesized as separately, the spikes are separately synthesized. So in the fourth step is the assembly of these synthesized particles. So the new viral particles are assembled from the genome copies and the viral protein. So these synthesized materials are assembled into a complete virus, and then these complete virus will be released out of the host cells. Right. Usually the host cell will be destroyed, and the new viral particles, thousands of viral particles will be released from a single host cell. And these newly released viral particles are what we call the virions. So you can see the diagrammatic representation of this process. This is the general pathway for viral replication. You can see the attachment. Then there is entry. Usually the viral chromosome enter into the cytoplasm. Then into the nucleus. You can see in this diagram the nucleus is being shown to enter into the viral chromosome is being shown to enter into the host nucleus. Then after that, you can see the viral chromosome undergoes transcription, translation, and also the replication, then assembly and release. In case of bacteriophages, or the viruses that infect bacteria, almost the similar mechanism occurs. There is usually, in case of bacteria, there are two possibilities. One is either the host chromosome will be lysed after the entry of the virus into the cytoplasm. In such cases, the virus are usually, we call them as lytic phages, and in some other occasions the entered viral chromosome will not lyse the bacterial chromosome. Instead, these viral chromosomes will be integrated into the bacterial chromosome. In that situation, these phages are called lysogenic phages, and the virus will multiply along with the bacterial chromosome, and each time the bacteria replicate, the virus also replicated, and a copy of the viral chromosome will be present inside the bacterial chromosome, and they'll be propagated. So these are the two possibilities you can see here. So in cases of these two pathways, you can see the basic pathways are the same. That means attachment, entry, all those things are there. And in case of lysogeny, even though the viral chromosome is there, which is not a permanent thing, because when certain unfavorable conditions are prevailing, this viral chromosome will get detached from the bacterial chromosomes, and these detached viral particle further will be turned into lytic cycle. So after that, there'll be cell lysis and release. So in bacteriophages, simply you can say there are two phases or two pathways for replication. One is lytic cycle, the other one is lysogenic cycle. In lytic cycle, the mechanisms are the same as already we discussed. There is entry, synthesis, then lysis and release. But in lysogeny, instead of initial lysis of the chromosome and the host cell, what actually the phase 2 is, the viral chromosome will be integrated into the chromosome and they propagate for long period, right? So all the, when the bacteria is taken, all the bacterial cell will have at least one viral chromosome particle, and these viral particles will be dissociated on occasion with some unfavorable condition like exposure to UV light and all those. And in that situation, this viral chromosome further will be converted into the lytic phase. So in that case, further lysis occurs. So this is the pathway in detail, the lytic phase or the lytic cycle. This phase, these viruses are otherwise also called virulent phases, virulent phages. You can see the mechanism. And this is the lysogenic phase, or it's otherwise called a temperate phase. And this temperate phase, you can see the lower part of the diagram. When the prophage exits from the chromosome, they will turn into the lytic cycle. So this is about the viral replication in brief.